evening, students. Welcome back to today's lesson. Today we are looking at cytology, and this is our lesson 2. And we are starting on cell organelles and their functions. In the previous video, we saw different cell organelles found in both plant and animal cells. And now today we are going to see each in details. So today, this is our lesson 2 of our cytology lessons. Remember, if you are new to this channel, you click on the subscribe button and then click on the bell that comes immediately after you have clicked on the subscribe button. So click subscribe so that you can get all our videos that we always upload. So now today we are on cell organelles and their functions. And as we saw in the previous video, we came to find out that an animal cell has very many parts when seen under an electron microscope, the one that we called the ultrastructure of an animal or a plant cell. So now today we are going to look at those structures which are seen only in the electron microscope, but if you check under the light microscope, you don't see most of those parts. So now an organelle is a mixture, an organelle is a minute structure within a plant or animal cell that carries out a particular function. So now most of you may be asking yourselves what an organelle is. So now an organelle is a minute structure. So now what is a minute structure? It is a very small structure, very small structure, and it carries out a particular function. So now some of the organelles which we are going to look at today, the first one is called endoplasmic reticulum, normally abbreviated as ER, endoplasmic reticulum. So now this endoplasmic reticulum, which you are going to understand very well, it is made up of two types. So endoplasmic reticulum is made up of two types. The first type is called rough endoplasmic reticulum. So now, we shall come to understand what rough endoplasmic reticulum means. And the second one is called smooth endoplasmic reticulum. So now, rough endoplasmic reticulum, it is where the ribosomes on its surface are abundant in cells which are rapidly growing or secreting enzymes. So now, in the diagram which we drew of a plant cell, you saw what we call the endoplasmic reticulum, and it is made up of rough endoplasmic reticulum and smooth. So, rough endoplasmic reticulum, it is the other one which has ribosomes at its surface. You find at its surface, for example, here, if this is your hand, then here its surface. If this one is the endoplasmic reticulum, we find here we have the ribosomes, the ones which you can, for example, call rashes. If you have rashes, so the rough endoplasmic reticulum, it has ribosomes at its surface, and it is commonly found in rapidly growing and secreting enzymes, those cells which are rapidly growing and the ones that secrete enzymes. So that's what this rough endoplasmic reticulum, it is normally found. And also they are made up of flattened cavities called cristae. So now you can see the diagram of a rough endoplasmic reticulum. So now on this diagram of this rough endoplasmic reticulum, we have some of the parts as we have said. So as you can see, here we have the ribosomes, these ones. So the ribosomes, they are these things which are on its surface, the ones that we call the ribosomes. Then the cristae, it is that inside part of the endoplasmic reticulum. Hope there we are together. So now we also have smooth endoplasmic reticulum. So now smooth endoplasmic reticulum For it, it lacks ribosomes on their what? On their surface. And they are most abundant in cells that produce lipid-related secretions, e.g., sebaceous glands in mammals, and the cell that secretes hormones. So now, for the case of smooth endoplasmic reticulum, it lacks these ribosomes on their surface. So the only difference between rough and smooth is that the rough ones, they have the ribosomes at their surface, and then the smooth endoplasmic reticulum does not have the ribosomes at the surface. Then the other difference is that the rough endoplasmic reticulum, for them they are found in rapidly growing cells and the ones that are secreting enzymes. But for the case of smooth endoplasmic reticulum, we are saying that they are found in lipid-related secretions, those cells that produce lipid-related secretions, e.g., the ones the sebaceous glands in mammals and the cells that secrete hormones. So the sebaceous glands in mammals, they are the ones that produce the oils on our skins, so that they can look oily. So those are the smooth endoplasmic reticulum. So now, what are the functions of this endoplasmic reticulum? How are they important to our bodies? So now, the first thing is that the rough endoplasmic reticulum, normally abbreviated as RER, their function, the first one is to associate and transport proteins synthesized on the ribosomes. So now, we shall come to find out that the major function of these ribosomes, their main function is to synthesize proteins. So now, when they are synthesizing proteins, because they are connected on these rough endoplasmic reticulum, they have the ribosomes on them attached. So they are there to transport the proteins that are manufactured there. And it is also important in cells that produce enzymes, e.g., cells in the wall of the gut. So the cells in the wall of the gut, they produce enzymes, and it is very important. Also, it conducts intracellular impulses, e.g., in muscle cells. Hope you studied coordination at your O level. So it conducts intracellular impulses. This one is part of coordination, which I think you covered in your O level. It also helps in attachment of ribosomes. So the ribosomes, it is where they attach. So it is an attachment. It also provides a large surface area for chemical reactions in a cell. So now we have the smooth endoplasmic reticulum, the one abbreviated as SER. So now this smooth endoplasmic reticulum is also important. The first thing is that it helps in producing lipids and steroids. Steroids, they also mean hormones. So now it helps in production of lipids and steroids. And steroids, they are the ones which mean the hormones. So now we can continue down. It helps in collecting and storing synthesized materials in a cell. Once materials have been synthesized in the cell, it acts as a store to them. Also, it helps to provide structural support to maintain the shape of the cell, e.g., in the retina of the eye. To provide structural support, to make sure that the retina in the eye is in its original position. It is the help of the smooth endoplasmic reticulum. So it is very, very important. Minus it, your retina would be shapeless. So the other function is that it helps in the detoxification process in the liver. So you know the functions of the liver. You must have looked at this one in your O level. So it helps in detoxification. So detoxification is the removal of waste products from the body. So helps in detoxification in the liver. And I think you know the functions of the liver. So now we can go on to the second part. We have seen the endoplasmic reticulum as an organelle that is very important majorly in transporting synthesized proteins by the ribosomes. So now let us check the ribosomes now. Well, remember, the ribosomes are attached on the endoplasmic reticulum. So now, how are the ribosomes? What is their function? So they are tiny organelles consisting of two subunits, one smaller than the other. So now, and remember, they are non-membranous. They don't have a membrane that bound around them. So now, they are tiny organelles and they consist of subunits, one smaller than the other. So now, as you can see from the drawing, you can see that the total size of this ribosome is 20 nanometer. Okay, 20 nanometer, and also it consists of the smaller subunit and the larger subunit. So this is the larger subunit, and also they are made up of equal parts of protein and RNA, ribosomal RNA. So they are made up of equal parts of proteins. Remember, the function is to synthesize proteins. Also, ribosomes are manufactured by what? They are manufactured by DNA in the nucleus, and they are very numerous. So now, these DNAs, they are manufactured by DNA. DNA in full stands for deoxyribonucleic acid. Deoxyribonucleic acid. So now, they are very numerous and they lack membranes that may be attached to the endoplasmic reticulum or may be free in the cytoplasm. So they can either be attached there in the cytoplasm or they can just be there free moving in the cytoplasm. But they are manufactured by the DNA in the nucleus of the cell. So that is the ribosomes. Also, ribosomes, they occur in both prokaryotic and eukaryotic cells. However, ribosomes of prokaryotes are distinctly smaller than those of eukaryotes. So now, I think you know the eukaryotic and prokaryotic animals. So the ribosomes in the prokaryotic animals, they are smaller than those in the eukaryotic animals, but all of them, they have ribosomes. Okay, all of them, they have ribosomes. So ribosomes also occur inside the mitochondria, even the chloroplast, and very many organelles. You find them there inside because their aim is to manufacture proteins. So they are found in very many parts. So now, what are the functions of the ribosomes? So the ribosomes, majorly, they have one major function which you have already looked at, and we are saying they are sites for protein synthesis in a cell. So their major aim is protein synthesis, manufacture of proteins within the cell. So now, also ribosomes, there are some ribosomes which lie free in the cytoplasm, and now they are sites for synthesis of proteins which are retained within the cell for intracellular use. So the ribosomes that are found lying free in the cytoplasm, they are just there in the cytoplasm, lying anywhere in the cytoplasm. For them, they manufacture proteins that are retained in the cell for intracellular use. But you have those ribosomes that are attached on the endoplasmic reticulum, as we saw. For them, they produce proteins that are secreted out of the cell, e.g. the digestive enzymes, hormones, and antibodies. So now, some of you may be wondering to see if these digestive enzymes are also proteins. So you check in the link in the description of this video. You will get a link describing the digestive enzymes, how they are formed. So you'll come to find out that they are proteins in nature. So the endoplasmic reticulum contains ribosomes. The ribosomes on the endoplasmic reticulum, for them, their main work is to produce proteins, and those proteins are always secreted out of the cell as digestive enzymes. But the ribosomes found in the cytoplasm, For them, they produce proteins that is retained within the cell for intracellular use. Okay, hope there we are together. So in case you have not understood anything about ribosomes, just write it in the comment section and they will have to give you the answer. Now we have what we call the Golgi apparatus. So now some of you may be asking yourselves what the Golgi apparatus is. So the Golgi apparatus, or the one that we call the Golgi body, or the one that we call the dictosome, it was named after the Italian scientist called Camillo Golgi. So now this man called Camillo Golgi is the one who discovered the Golgi apparatus, and therefore he named it after his name. That's why it is called Golgi apparatus. So that scientist was called Camillo Golgi. So now you may be wondering, you may be asking yourself, what is Golgi apparatus? What is it? So now the Golgi apparatus is similar in structure to the smooth endoplasmic reticulum. In structure they are the same, but it is composed of a membrane-bound disc-like cavities pulled up with secretory vesicle banding off. So what are they saying? This is the Golgi body. It has those secretory vesicles that secrete different secretions. So that is the only difference. But for the case of the endoplasmic reticulum, they don't have it. So now you can see how it is formed. We also have the functions of the Golgi body. What does the Golgi body do? What is its main function? We also need to understand that thing. So now the Golgi apparatus is well developed, especially in the secretory cells, e.g. in the pancreas and the salivary gland. So it is commonly found. Where is it commonly found? It is commonly found in the pancreas and the salivary gland. So it is found in the pancreas and salivary gland because those ones, they secrete different secretions. That's why they contain much Golgi body. So now the first function of the Golgi body is production of glycoproteins, e.g. mucin, in the process called the glycosylation. So now you may be asking yourself what glycosylation is. So now glycosylation, it is when a carbonate and a protein, when they combine to form a glycoprotein. A protein combining with a carbohydrate to form a glycoprotein. So that process is done, which we call glycosylation. So production of glycoproteins, it is done by this Golgi body. So now we also have that the Golgi body contains enzymes for the synthesizing of the glycoproteins. That one I have already seen it. So it contains enzymes which manufacture the glycoprotein. So its major function is to manufacture the glycoprotein. Also we have production of secretory enzymes, e.g. the digestive enzyme and the pancreas and other parts of the digestive system. So we have production of secretory enzymes, those enzymes that are used that produce secretions, e.g. the digestive enzymes and of the pancreas and other parts of the digestive system. So now most enzymes, they come due to the Golgi body. So the Golgi body manufactures different enzymes, e.g. the ones from the pancreas, the pancreatic amylase, the pancreatic lipase. All of those ones, they are due to the Golgi body. Also we have formation of lysosomes. So now formation of lysosomes. So don't worry about lysosomes, you're coming to them and you see what are they, because they are also found in the cells. We shall understand them. Also secreting carbohydrates such as those involved in the production of new cell wall and the insect cuticles. So it helps in secreting carbohydrates, like those ones used in production of new cells. So without the Golgi body, new cells can't be formed because carbohydrates are needed in the formation of cells. And the Golgi body, they are the ones which form those cells, those carbohydrates that are going to be used in making the cell. Therefore, what does that one mean? It means that if the Gorg apparatus is not available, then carbohydrates won't be manufactured. The glycoprotein would not be available because the carbohydrates from nowhere, where can they come from without the Golgi body? So glycoproteins cannot also become available if the Golgi body is missing. So now, incidental repressioning or renewing of the cell surface membrane, that is to say, when secretory vesicles empty their content the exterior of their membrane, they fuse and become part of the cell surface membrane. So this one is like a note that when the secretory vesicles they empty their content, when they are emptying them out, they come and join the wall, the cell surface membrane. Either if it is an animal cell, they join the membrane. There's a membrane outside one, the one that we call the cell membrane. So now we can come to see what the lysosomes are, which you have seen that you're going to understand very well. So now, lysosomes, first of all, where does that lysosome come from? So that lysosome, it just comes from two Greek words. So some of these biology words, they become very hard because they are Greek words. So now, they come from two Greek words. Lysis meaning splitting, and stomach, which means body. So which means splitting body. But now, what does splitting body mean? Don't worry, we're going to understand. They are dark spherical bodies bound a single membrane. They have a single membrane. Okay. Also, the lysosomes, they contain digestive or hydrolytic enzymes. They contain digestive enzymes, which you're going to see down here. Also, they are physically separated because they can destroy cell contents. They are physically separated. Why? Because they can destroy cell contents. So take note of that. They can destroy the cell contents, but we shall understand how do they destroy the cell contents. For example, if a cell is attacked by virus, how does it come to destroy that virus? We shall come to understand its function very well. So these are some of the enzymes found in the lysosome. We have lipase, which works on lipids, glycosidase, which works on polysaccharides, protease, which works on proteins. We also have phosphatase, and phosphatase works on proteins. Okay. We also have DNase, which works on nucleic acids. So lipase works on lipids, glycosidase works on polysaccharides. So now, having looked at those enzymes, we need also to know that lysosomes are most abundant in cells with high phagocytic activity. So they are found in those cells which their main function is high phagocytic. Phagocytic means they go and engulf. If a virus comes, they engulf it, swallow it, then they go and die. Also swallowing, they swallow it or engulf. Such as things like the white blood cells fight different viruses. So that is where they are found. But now, there are other functions of the lysosomes which they do in carrying out these different functions. For example, they can be used in chasing away viruses. Now, for example, if a virus like COVID-19 is in you, it has entered in you, for example, you are sick, how do the lysosomes react towards it? We shall answer that question in our next video. So, thanks for watching today this video, and we have seen very many things. Also, this is our lesson 2. Remember, lesson 3 is coming next week. Don't miss. Share with your friends, and please, if it is your first time, subscribe onto this YouTube channel so that you can continue learning here when you are just at home. So I remain Kamate Ntuyenabo. We meet in our next lesson. Thanks very much.